Good morning. Namaste. This is Dr. Uma Vital, Faculty of Agriculture and Veterinary Science, Jayoti Vidyapith Women's University, Jaipur. And I'll be continuing with the lecture on basics of immunology. So in the last session, we had covered the history of immunology, what the concept of immunity was, and such details. Now, in this session, we will be covering what is immunity and the two and a major concept in immunity, the humoral and cellular systems of immunity. So, what is immunity? Immunity is our natural defense mechanism, and it is comparable to our real life defense mechanisms. Like if you have a house, there will be a wall, and so that wall becomes one line of defense. Now you have a watchman over there, then that becomes the second line of defense. And then after that, again, if that watchman is unable to prevent from someone entering the house, then they could always signal to another watchman or some person inside, and they could also come and try to resist that person. So uh, they could get as much support as needed in order to get this person out of the house. So in the same way, when an unwanted entity, a pathogen that can cause an infection or can drain the resources of our bodies trying to enter our body, then our body will need protection, and that protection is known as immunity. So if we have a security guard at the entry, so something similar, like there are antigen presenting cells or APC in the body that will present the antigen to other cellular immune systems and then get rid of those. And then the second line of defense will be from the B and T lymphocytes, and then they can also, in multiple ways, counteract the entry of the pathogen or its establishment inside the body, and thereby remove it from the body entirely. And in a similar way, if there is a fight between these the intruder and the bodies or the house's security system, then what happens? There might be some damage that is happening, and that is something akin to what happens in a hypersensitivity reaction, that what is perceived as the intruder, the body will attack it, but sometimes there will be collateral damage to the body itself, and that is a hypersensitivity reaction. Now, in the same way, if we have kept a security guard, but that security guard himself or herself is corrupt, and she is or he is trying to do harm to the body, then that is typical of an autoimmune response, that our body's immune system is not doing its duty, but it's actually uh, harming our own body. Then that becomes the autoimmune response or the autoimmune diseases where the immune system is defective. So now coming to the definition of immunology, it is defined as the study of molecules, cells, organs, and the system responsible for the recognition and disposal of non-self. That will be whatever is external to the body or foreign substances. And the function of the immune system is to recognize this non-self from the self and to defend the body from the foreign substances. Immunology has got multiple sections, but one of the major sections is immunity. Is the adaptive response to foreign substances or antigens, which could cause infections. And immunochemistry is the study of the nature of these antigens and our body's response to these antigens. That will be the antibodies, the complement system. And immunobiology is the study of the activity of the immune cells as such, like the B lymphocytes, the T lymphocytes, the antigen presenting cells, mast cells. There are different types of cells that are involved in the immune system, and immunobiology deals with the study of all these cells. Uh, and Immunology is also one of the most developing medical sciences because it deals directly with human health, and most of the research is uh, focused on either improving human comfort or improving human health. So immunity is the field of human health. So that will be one of the major focus of research always, and thereby it has got implications in immunopathology, that will be with pathology, pathogens, tumor immunology, that will be with cancer, immunopharmacology, that is how much drug is needed and at how many times a day, how should you have it, before food, after food, what is the better way to have it. Transplantation, like if we are keeping a foreign tissue or an organ in our body, how to avoid our body destroying it by identifying it as non-self. And then the immunological disorders like HIV and autoimmune disorders, all that will come. So coming to the main topic of discussion, humoral immunity and cell-mediated immunity. So humoral immunity is an antibody-mediated response that occurs when the foreign material, like antigens, for any foreign material is defined as an antigen. There are incomplete antigens as well. They are known as hapten, but they will be covered later. So they are detected in the body. And this foreign material typically includes extracellular invaders such as bacteria. The humoral immunity is primarily driven by B cell lymphocytes. So what happens is there are naive B cells that do not have any memory, that have not been exposed to any antigen so far, and they circulate throughout the body in the lymphatic system. And these lymphocytes express a variety of antigen-specific molecules that are essential for the detection of infectious agents in the human body. And when the naive B cells encounter an antigen, they will undergo differentiation process to create memory B cells and effector B cells, and effector B cells will produce the antibody that will work against the particular antigen, and then that will lead to a immune response. So there is also the help of the T lymphocytes in this process. Like T lymphocytes are activated by something known as major histocompatibility complex class 2 receptors. So MHC, major histocompatibility complex, this plays a very major role in the activation of T cell lymphocytes. And they are associated with microbial associated antigens, and the activated memory B cells express these antigen-specific molecules and their surface, while effector B cells secrete these molecules to the blood, and it binds to the antigen of interest. But so I've been talking about this, that B cells are secreting antibodies. But what is an antibody? So antibodies are heavy proteins that are approximately about 10 nanometers in size, and they are produced by B cells, and they have specific antigen binding sites that can bind to very specific antigens. Like you have one antibody doesn't mean that it can bind to all kinds of antigens. Antibodies are very specific in that they recognize very specific particular sequences of antigens, and they can bind only to them. And once these antibodies effectively bind to the target antigen, then they can either neutralize the target antigen, or they can recruit other immune cells such as phagocytes, which can further remove the antigen from the body by phagocytosis and such processes. Now, each B cell has its own set of antibodies with unique antigen-specific binding sites. So when an antigen matching the antigen binding site binds to the nave of memory B cell, then it activates the B cell to produce more antigen specific antibodies. And once that is happening, there will be enough antibody in the cell to counteract the attack by the antigen. So uh, there are like different functions as defensive molecules with for these antibodies. So these functions are neutralization of infectious agents via blocking or antibody dependent cellular cytotoxicity. Then there is activation of the complement system, which leads to complement dependent cytotoxicity. And then there is the binding of foreign substances to be destroyed. That is a process known as opsonization that will lead to phagocytosis by the phagocytotic cells. So antibodies can also participate in processes that lead to lysis or the killing of the cells. And that is what is through the complement cascade, or that complement cascade will involve a lot of cytokines and other chemicals. And antibodies that coat pathogens, that is known as opsonization. So once the antigen has been opsonized, they can be phagocytosed by the phagocytotic cells of the immune system, and thereby they can be destroyed in the lysosomes of these phagocytotic cells. So that is the part about humoral response, which involves the antibodies. Now, what is cell-mediated immunity? So I have told that in humoral immunity, B cells are involved, and these B cells will produce antibodies. Now, in cell-mediated immunity, actually the cells themselves are involved. So there will be T cells, as I had discussed earlier. The T cells are involved in cell-mediated immunity, and they need antigen presenting cells that have membrane bound MHC class 1 or class 2 proteins. MHC class 2 is actually involved in activation of B cells, and MHC class 1 is involved in the activation of T cells. Now, when antigen presenting cells having MHC class 1 proteins present these T cells to present the antigens to T cells, then they will differentiate. They are also named T cells. So they will differentiate into helper T cells or killer T cells, and the cell-mediated immunity will come into play at body sites where cells are infected by virus, bacteria, fungi, and actually T cells and the cell-mediated immunity is the response to cancer cells as well. So what lymphocytes are involved in the cell-mediated immunity? So it will be helper T cells and killer T cells and macrophages. Macrophages are the primary phagocytotic cells in the body. And when a helper T cell finds an antigen presenting cell in the body, it releases a set of signaling proteins called cytokines. And these cytokines will activate the killer T cells and macrophages to they are essentially recruiting these killer T cells and macrophages to the site where the antigen has been detected, and then then they will destroy the antigen that has been bound to the antigen presenting cells. So it is not as if like if you find an antigen, then they will be taken somewhere else and then destroyed. The body's first response is to recruit the cells to this site so that there is no chance of the antigen spreading to other parts of the body. Now, what is so I would like to summarize that what is the difference between humoral and cell-mediated immunity. So in humoral immunity, it is an antibody-mediated response, whereas in cell-mediated immunity, it will be a T-cell-mediated response. The site of activity for humoral response is extracellular fluid, such whereas cell-mediated immunity, the these T cells and macrophages are recruited to the location where the antigen presenting cells are present with the bound to the antigen. So the main cell types involved are B cells in humoral immunity and T cells in cell mediated immunity. And humoral immunity is quite fast, as in it responds very fast upon detection, whereas T cell, the cell mediated immunity is much slower. And the antigen type is mostly extracellular pathogens like bacteria in case of humoral immunity, whereas it's intracellular pathogens and cancer cells, which are the major types identified by the cell mediated immunity. And the major histocompatibility complex proteins involved are MHC class 2 proteins, as I had said, for humoral, and MHC class 1 proteins for cell-mediated immunity. So that will be that for humoral and cell-mediated immunity. Now, in the next session, we will be covering the innate and acquired immunity. That is again a major classification for the immune responses or of our body. Have a good day. Thank you.